This keynote is sponsored by BlackRock, so thank you, BlackRock, for sponsoring. It's my honor, pleasure, and interest to announce our speaker, Karunesh Ganguly. Karunesh is an MD, PhD neurologist and neuroscientist at UCSF. He in the San Francisco VA Medical Center. He completed his MD, PhD in the University of California, San Diego. Subsequently, did his residency at UCSF. And during his residency, worked as a postdoc with Jose Carmina, and got into BCIs, and has published some seminal works in the field in terms of looking at BCI control and what's happening to the neurons when these BCIs are being controlled. In addition, he's a triple threat. He's a clinician, scientist, and teacher, obviously. In terms of his research, not only does he do BCI work, but he's got some amazing basic neuroscience work, including looking at motor function, motor learning, and learning during sleep. So, but today he's mostly going to be telling us about his, I assume, his work with BCIs. So give it up for Karunesh.
Thanks, Mark, for that kind introduction. And yeah, I'm going to limit it to translational BCI and one slide of indulgence on basic science. So today the goal is, I'm going to talk about our towards the goal of BCI robotic control in the home environment. And just to set expectation, we're not at the goal, but it's towards the goal. And what I'll show is our roadmap in our clinical trial towards making BCI robotic control potentially viable in a complex home environment. And of course, part of what motivates all of us, I think, is try to reduce disability in the world. And I'm from the US, and certainly in the US as well. And this figure comes from the Christopher and Dana Reeve Foundation that did one of the most comprehensive surveys of people living with paralysis in the US alone. And you can see how large it is. And of course, BCI in its current form will probably help a portion of this, but we all hope to broaden it to other indications. The other amazing thing that probably most people are aware of is how quickly BCIs have moved, but it has a long history, right? And particularly wanted to shout out this Fetz 1969, a solo science author with paper, which is amazing. And if those who haven't read it, it's really foundational for a lot of what's going on. But certainly it's been exponential with the translational work. And regarding that, of course, you all know that there's been rapid and remarkable proof of principle in the last 1 to 2 decades. This includes cursor control, typing, robots in paralyzed patients. And because what I'll talk about is robotic control, wanted to highlight, of course, the work of Lee Hochberg, Jen Collinger and colleagues, and wanted to point out, some of this meeting has talked about COAs, clinical outcome assessments, and it's worth pointing out the Collinger paper here, they had an ARAT increase of 15, which is pretty amazing. It's 3 times the minimal clinical important difference. So it shows if we can move these robot controls into the home environment, huge potential for functional gains in very meaningful scales that are well accepted clinically. And so today, really focusing on this goal of how do we take all this great work and try to move it towards something that could be useful in a daily setting. And so how might we do this? And this has been brought up a lot, critically participant feedback, really focusing on tasks that matter for people, and ultimately testing in complex environments that mimic the home environment, and ultimately, of course, the home environment. And we can learn lessons from other fields. So these statements here are from review papers looking at prosthetic use. These are non-BCI necessarily peripheral prosthetics, and importantly, it's been repeatedly found that the level of function has to be important and be meaningful per patient, right? Without that, it just sits in a drawer, has to have an acceptable form factor, and importantly, and this is something we think about a lot, has to be simple to use, which involves minimal calibration, setup time. You can't have engineers run around behind people. And so these are clearly lessons that people have documented over and over again. And so what are some of the challenges for motor BCIs? And I put motor, of course, because cursors can also help with communication. And so some of the things we think about are stability, calibration, making control simple, intuitive, And I'll show you some. We have been testing sort of dual paradigms back and forth to see which are intuitive for participants. Enable control that's useful for daily activities. This, of course, requires feedback, control in natural environments, and something I'll talk about more, and we've been focusing a lot of this internal states, right? So when we look at a BCI control, it's an external thing we can observe, but we find very interesting changes in internal states during control that strongly predict fluctuations in accuracy and errors that will become really important, I think, for real-world control. And so just to think about, well, and this is, of course, in comparison to with the speech prosthetics, which have moved really quickly. So one of the questions is why our BCI robotic control is much harder, right? And one thing is, right, we're, at least in this form, and maybe FES will be different, exoskeletons will be different, but if it's an external artificial actuator, this has complex dynamics, it's non-intuitive, it's not natural, you know, and then when you're, and I'll talk about this, like the really hard part is object interactions that are, you know, natural limbs, so there's so much feedback, so many processes that make it really hard for robotics, and we need to think about that. Planning, execution, movement is not just about moving, it's about environmental scanning, prediction, I'll talk about that, feedback, and so there's just a lot of things that we have to recapitulate. And it's important to think of them as how does the intact nervous system do this, right? And so one critical thing I think that's been known for decades, and I'll talk about this in the idea of affordances, so right, when we move, before we move, our brain through very sophisticated pathways have estimated affordances for objects, and there's models, internal models of that, and we need to think about that for complex control. Not to mention that, right, most of the prosthetics we talk about are using cortical signals, and there's unbelievably sophisticated evolutionary conserved mechanisms, for example, the basal ganglia that help with automation, control. I can speak and wave my hand at the same time effortlessly. Brainstem areas, for example, the reticular formation spinal cord that really help with constraining the degrees of freedom and robustness. And so so this is something we think a lot about. How do we, from cortical signals, think about these things that are all nested within a very complicated evolutionary conserved nervous system? And so then today, just focusing on what I will try to outline today, and these will be in three main topics. One is tracking representational instability to allow stable decoders. So this is trying to address making control simple, reliable over long periods. Number two is this unpublished work in preparation where finding that there's slow fluctuations in internal states that predict inaccuracies and how to use that to improve control. And last but not least, but this idea of supervised autonomy, which is more sophisticated machine learning. Kind of think about what large language models do for speech prosthetics. How can we use computer vision, other assist frameworks to enable complex control? So to start with, I want to just, you know, we're influenced by our history of our training and so forth. Start with some of the work that have really influenced the work and how we approach it. And this was work, as Mark alluded to, was when I was a postdoctoral fellow at UC Berkeley in the Carmena lab. And we were doing cursor control at the time in monkeys. And this was recording spikes from premotor, motor cortex, and using linear decoders to look at cursor control. Fairly straightforward now. And all the monkey had to do was just do a center out task. And one of the things that really was disappointing, we'd spend an hour calibrating, get really good control, come back 2 days later, it just didn't work, right? And we have to redo this all. And so that kind of just set some of the questions in my mind of like, why is that? And why is there instability? And so some of the lessons I learned back then was signal stability is fundamental, right? So these are hard experiments, so it's hard to track spikes over time. But in short windows, especially if monkeys have been implanted for a year or two, you can get stable windows. And so these are the units. You can see that waveforms are super stable for like 20 days or so, right? And there, the monkey was just doing cursor control. And what was interesting was, despite reasonably good control day to day, especially if you look at their learning curves by the end of the session, that the neural properties changed over time. So if we look at day 3, these are the tuning properties of the neuron, and this just reflects, the center out is a circle, and this is just the direction of modulation that the neurons care about. So you can create a map on day 3. When we now look at day 14, we think they're the same neurons, everything's the same, but the map has drifted. When we look at day 18, the map has stabilized. And what this correlation is a pairwise 19 by 19 matrix, where it's essentially just a correlation of these maps. And interestingly, the first 5 to 7 days, you can see that the correlation's low, and then it snaps into something much larger, and then you get a value of roughly 0.8 to 0.9 throughout, right? So this again says that there are mechanisms that ultimately lead to stability, but there's instability. And again, one indulgence for basic science to move to translation. And for a big part of our lab has been trying to understand this in animal models. And I wanted to give you some clues and move to the robotic work. And one is that we've found through sleep that the nervous system is always trying to balance remembering versus forgetting. There's really, and we can actually manipulate to make animals learn faster or slower. And so just to point out that these learning rates are not something that's just, it's not trivial. There's a lot of mechanisms to do with credit assignment and so forth. The other is that this learning process here, these first 5 to 7 days, seems to be not just a motor cortex thing, it's involving hippocampus, prefrontal cortex. And just to give you a hint of that, so in this study, we looked at hippocampus, medial prefrontal cortex, and motor cortex throughout our entire learning process, right? And it's very striking. In the first 5 to 7 days, depending on the animal, the manifold, if you will, and Juan talked beautifully about this, is very unstable. Like, you don't get these beautiful rotational dynamics. It's just all over the place, and this is reflected by the fidelity. And then all of a sudden, when the task becomes predictable, the animal can basically, there's no more further increases in accuracy. The system switches to another mode where hippocampus disengages, prefrontal and M1 start to couple more. And you get this really reliable pattern. And so just to point out that these instabilities are probably a feature, not a bug. It's helping the nervous system set in policies that are more rigid, less flexible, right? And last but not least, there's a lot of other interesting thing, and a big part of our basic focus is on rest periods. So a lot of the processing we find is actually not during learning itself, but during the breaks itself. But anyways, so moving on, but just to give you a sense that this instability that I'll show you in the translational work may be something related. Of course, we don't know in human participants, but just to give a flavor that this isn't just noise. There may be a feature here. And so then, with this drive to say, okay, we need to have more stable signals, and of course, prior to us doing this, there's been a lot of work showing that ECOG may be more stable and has sufficient decoding ability. And together with Eddie Chang, which I'm sure all you know has done great work in speech prosthetics, and Adlin Tuchan, who's also a neurologist at UCSF, we started the Bravo trial, and it took us about 4 years to get the IDE, and then we launched the trial in about 2019. And I wanted to shout out, because Mariska brought up the community. This wouldn't have worked without BlackRock and Rob Franklin really helping us with the IDE, PMT, and Canova. And the 4 years was basically getting 3 corporations to open up their FDA folders and do a lot of, so it was interesting, and ultimately we got it through, and it's been incredible. But just to point out, there's a long path to this. It wasn't so simple. And then I'll highlight the people in lab that did the work as I move on with this. So today I'll just talk about 2 of our participants that was involved in what I'll show you here. And first one is Bravo 1, and I'll just use this moniker Bravo 1, Bravo 3 as the 2 participants. And both have similar lived experiences. So Bravo 1 is a 40-year-old man who had a vertebral dissection, had a brainstem stroke, and is anarthric, severely dysarthric, doesn't really make functional speech, and has spastic tetraparesis. He has residual wrist function, so he can, that's his only function on his right wrist, so he can move a wheelchair around, but that's the extent of it. And then Bravo 3 is a 47-year-old woman, also with a brainstem stroke. It's thought to be she was on contraceptives, also had a basilar thrombosis, and resulted in anarthria and spastic tetraparesis. So these were at least very similar lived experiences, phenotypes. So it just gives us a way to at least settle on some common parameters for developing a system. And I should mention, this is, of course, a FDA investigational device under an IDE. So before I move into robots, we did one first study with these participants, or at least one participant, Bravo 1, and learned some lessons that will be relevant to the robotic control. And here is, again, cursor control, the usual center out. And we were interested in 2 ways of thinking about the decoder, right? So one is a daily initialization, which is in the peach. So every day we'd create a new decoder, and we'd have the cursor move around in a stereotype way. The participant would imagine. We work with them to figure out the right actions to imagine. And then every day we'd do that. In this lighter blue is when we took one decoder, trained it on one day, and just kept that weights throughout. And kept optimizing the weights even though they were 3 days apart or 10 days apart, so no recalibration. Interestingly, when we do the daily initialization, as you might expect, there's a lot of variability. And here, we record performance from, every day we did 1 hour of calibration, and then all the recording, the performance here records the rest of the time. There's no, let's find the best time or not. This is all performance. And as you might imagine, there's a lot of variability with daily training. And interestingly, I think from a learning perspective, if you look at the weights here, so these are the decoder weights. These are undergoing a closed-loop adaptation on a single trial. This was a method developed in the Carmena lab using a Kalman filter adaptation method. Interestingly, with this learning in a given day, there's convergence to some weights. When we reset it, when you look at the dot product between them, they're orthogonal. So every day there's a reset. The weights are essentially resetting in a way that may be unnatural. And this may be one of the problems with converging onto some neural representation. Contrast that with now if we take the same weights and day after day continue the adaptation for about a week here, about 7 days total, but spread out over time just for clinical reasons. And there we see that the decoder kind of converges over periods of time to something more stable. And we get very stable and increasingly complex function. Again, suggesting that there may be some across-state plasticity that might be kicking in when we do this. And another thing to point out is that, one of the things that was surprising to us, how unstable visual imagination is, right? So, when participants are asked to imagine actions and imagine what they're doing, so it requires a lot more thought to get that to stabilize. So what this shows is how good is our model in an offline sense during these days of there's the cursors moving around and the participants are imagining what to do. And what we find is that actually the models on any given day, R-squared cross-validated, are quite good, meaning that it can actually predict the cursor. But if we add more and more days of data, it actually collapses, right? So it says that the model cannot converge as you do more and more days, which again we interpret to mean that the neuron or the neural response to the observed observations not very reliable. This may be why fixing a model and decoding slowly is better. And this, when we did this, when we now fix the decoder, we were able to get greater than 30 days. And I'll use this term plug and play, meaning a decoder that doesn't need adaptation. Super simple. We plug them in, do a baseline for 2 minutes, and it's good to go from then on. And we in fact find that when we have a plug and play decoder, even though firing rates will change or global firing rates might change, it's very robust. It doesn't need, it's not brittle to local changes, and I'll show that a little bit more. So then moving forward with robotic control, right? So this is something we spent it's a 3-year study to really understand different control types and how to enable long-term control of a physical actuator, and this was the work of Niklesh Natraj, and then Sarah Seiko played a really critical role in the robotic parts. And so these, again, same participants, and I wanted to point out B1 has 128 channels, B3 has 253, so same footprint overall, but just higher density. And we look at some outcome measures of robotic controls I'll show you. And just to set expectations, right, there's obviously a lot of actuators out there, and we focused on this Kinova J-code because it's extremely robust. It's actually used in the community by people. There are lots of YouTube videos of people using joysticks and so forth. And with discussion with our participants, they like this device, and it's actually wheelchair mounted, so it's something that's relatively low profile. And for them, this was what they picked, and that's what we will focus on as actuator. And wanted to point out, like, a natural question is, why is this slow, right? We'll see the movements are somewhat slow. Number one, the FDA limits speed. Number two is our participants actually prefer slow but accurate over anything fast, right? So this is worth keeping in mind. And this is a great quote from B1 because he shows his videos to his friends, and they're like, why is it so slow? And he has this thing, please excuse my slowness, I value what I got. So this is just to say these are participant centered control parameters that we arrive on. So part of this, we wanted to next think about like what are the ways to control a robot, and towards that we went through and tested a slew of imagined and attempted movements, and these are just without a robot, just in a setting where we show them body parts and say imagine this, and we do this over a few days, and we were able to decode quite robustly a whole range of actions, maybe not shockingly, right effectors, left effectors, the entire body, head. I only show a few of them, but we did a very extensive mapping. This is not a confusion matrix, it's actually a distance measure. So this is a Mahalanobis distance, so which is just a way of thinking of it as d prime, which is distance in the high dimensional space normalized by the variances. So it just gives you a relationship of this, and these are highly significant. And in the paper, we do statistical analysis say these are, 99% of these are true statistically valid relationships. And so it just gives a sense that we can decode quite a lot of fragments of movements, right? And the question becomes, how do we map them onto continuous robotic control? And to do that, you could imagine two strategies, right? There's more. And so what I showed you in the cursor control was continuous control, basically a regression of the neural activity to cursor position along with the noise model that the common filter had. So we tried this continuous model, and then an alternative is this what we call IBID. It's input-based integrated control, and what it is that the imagined actions, and I'll show you what they are basically velocity impulses to the robot, and then the robot moves. These are all position control, not impedance control. And what we get are, and you wouldn't be surprised by this, when you have a robust action that a participant can do, you get very nice trajectories in the cardinal axis. And of course, they can also go off axis, but this is what we show here. But when we do the continuous regression, and we do this across days, this IBID framework, as I'll show you, converges onto something very stable, very flexible. The continuous did not. We kept going with that. It just had a hard time converging. And participants, right, we want to go with IBID, not the continuous. So this is just our experience with our participants. And to give you a flavor of why, that if you want to control something, you want signals that are separable, right? You'd rather decode something like this than something like this, right? And what these are we're taking the high dimensional space, which is like 128 times 3 features, or 253 times 3 features, building a autoencoder, and so this is a 2D latent space, a manifold, just for visualization, but all the analysis is done in the full high dimensional space. But just as a visualization, you can see with just some closed loop decoding, these imagined actions get just separable, so it's very easy to decode. Whereas when the continuous, they just keep getting jumbled, and the participant and the brain seems to some version of, they're having a hard time really separating them, and that may be why the trajectories are noisy. We didn't continue with this, so it's very possible continuous decoding a future thing, but for now we just focus on this just to think practically. And so the way we build this up is to take, so the Canova has 7 degrees of freedom, and I'll show you the hand control. So we first focus on just transport endpoint XYZ control. And we did this by mapping the action states onto the endpoint. And this is essentially a joystick, right? And this gives you a sense, with a couple of days, even though they're somewhat artificial in the mapping, that they get really good at invoking them when needed. So this is a pi bullet environment of a Canova robot. And what's being shown to the participants are go down, go up, go left. And with a little bit of training, they're really good at this. So, and if you ask them, what are you doing? The imagined action's not what they're imagining now. It's just I'm going up or down. So it becomes extremely natural very quickly. And this just gives you how robust they are. And for a lot of reasons that we've been using very simple machine learning. This is actually with the perceptron, because they're easy to update, not data hungry. And then in the future work with the robot, we did use the LSTM because it has a history built into it. This has no sense of time. It's an immediate classifier. But you can get a sense of how stable it is. And these are some of the single trial responses. And so you can see that some of the actions are extremely robust in the hand knob, right? So this is a clinical ECOG. It's even better with the 253. I'll show you that. And then, so this is a confusion matrix of 12 of the actions. And then, they can be mapped to discrete things. So this is a selection task. I don't know if it's going or not. That's a bummer. If no videos work, oh, there we go. So you can see that can be continuously mapped, or they can be actions. And so then we map these onto the Canova robot. And here, just for simplicity for now, and we're working on more complex control schemes, we're using the same 7 degrees of freedom, and they're mapping to the hand. So, and this is something Canova that's their control strategy, so we kept with that. So it's a transport in XYZ, and then they have a mode switch with another button, and then they switch to a control that's more end effector based. Again, very natural. You would think this is kludgy, but these 7 states, I think it's a testament to their flexibility that they don't get confused by them, and we've tested it, not very cognitively challenging for them. And so this just shows you the type of control. And then part of what I wanted to highlight was that there's instability, and that's what really helps us get to tracking that, helps us get to long-term stable control. So this is, again, I've shown you this. I want to go a little bit into this detail. So these are 10 days apart, and in these experiments, we're doing them daily, morning session, afternoon session, so we've got a lot of data. And so if you look at a day 1 manifold, these are just open loop. There's no feedback. They imagine thumb, middle thumb, head, leg, and so forth. And then the closed loop is after about a 10-minute block. We realign to the ground truth label, update the decoder, do another one, and that's what CL2 is, closed loop decoding, and we get this really nice separation, right? We can do this every day, and control's quite good every single day, every session when we redo this, and that you can see day 10 manifold looks pretty good, right? But then the problem becomes that we find that it doesn't generalize across days. It's an interesting phenomenon where we can plug the participant in the morning, afternoon, really robust. Day 2, it degrades. By day 3, it's really quite bad. It doesn't really work too well. And so the question is why, and how do we use that for long-term control? So one way to think about this is, again, separability, right? You want actions to be separable in order to decode them better, right? It's fairly straightforward. And so here is an example of if we build a latent space on day 1, again, this is a three-layer autoencoder at the bottleneck, so it's just a way to visualize. If we plot day 10 into it, it actually doesn't look very good. It's at the origin, right? And so it says that that day 1 manifold doesn't generalize to day 10. If we now sample all 9 days, day 10, maybe it's not surprising, but day 10 actually looks pretty good. So it says that there's some representational drift over time. If we sample, then it gets to be a really good decoder. And we did this in a systematic way, and we find that in both participants, like 9 days, 8 is even 7 to 8 is sufficient. That there's it's across days. So if you had 1,000 trials, sampling the same day wouldn't help. You'd rather do 100 across days, and that's what we found. That this really leads to, as I'll show you, very stable decoders over at least a month. And it's worth pointing out that every given day we can get good performance. This is just about how do you get stable long-term performance. And what this shows here is that if we use latent spaces that sample more days, like day 1, 1, 2, 1, 3, and so forth, by the time we get to day 1 to 8, you can see it starts to saturate. And we have a really nice relationship between separability in this Mahalanobis distance with control, if that makes sense. And so again, the gist of that as take-home messages, and that the representations are pretty stable within each other, so their relationship is stable. Like these clouds look relatively stable relative to each other, but their centroids are moving around, and we need to track them for stable performance. Just to give you an intuition of this is sort of a more machine learning, but a cool visualization, where, so, if you want to build a decoder, this is based on real data, so this is based on a manifold that's sampled all days, right, all 10 days. And the goal is to separate these 2 clouds of real data. These are just based on variance. And so if we now, these are all day 10 data clouds, right? And ultimately, you want to make a decision boundary that looks like this, right? And so it's interesting if you track what's going on with the decision boundary as you add more days. So the day 1 decision boundary with day 10 is horrible, right? It just doesn't see the difference. As we add more and more days, you can see how the boundary starts to converge onto something stable. And as I was saying, by day 8, it's actually pretty good. And maybe 10 is overkill. But you can see how it converges onto something. And why this is important, we think, is A, stable performance across days, but also within a session, right? Like this decoder has seen massive drift. So tiny drift, it doesn't care about. So it really leads to very robust within session and across session performance. And so then what was our outcome measure for stability? We did it 2 ways. So one is this, what we just call a joystick task, where the robot doesn't move, and they have to just engage the right action. And so you can see the accuracy is pretty stable, and there's no statistical change across time. And we did this in B1 in 2 experiments. One is pre-COVID, one is when COVID we opened up again, so about a year later, and we saw the same thing. And in B3, the same thing. So again, we get very stable performance, and the time to target is in a more dynamic task where they actually have to move this is a virtual task to an effector. So again, this says that once built, we can get at least 40 days of stability, right? And this is, again, plug and play. We just do a baseline each morning, and that's it. And then, of course, we moved this to the physical robot, and here we worked on 2 outcome measures. One is a rotate task, where they have to just pick up a block, rotate it, and drop it, and we test this day after day. And Bravo 1 particularly liked this one, so he was like, I'm not bored by that, so I can do it for, as part of the routine. Oh, did it go away? Huh. Should I read? Oh, there we go. I won't touch anything.
Yeah, make sure the HDMI is good.
Okay. All right. I'll just put this back on. So we had these 2 things that we measured over and over again, which is this wall place task, lift it up and put it up. And there's some variation, but these are fairly challenging tasks, so we get pretty stable. One thing to point out, it's not forever, right? So if you keep tracking this, and we did this for about 7 months, that there is a degradation of performance by day about 40 to 50. And so then we've been tracking this to do about an 8.5-minute calibration. We can go back to 100%, right? So these drops are based on pure plug and play, but fairly brief calibrations can bring it up without. And this is just basically, and because we use very simple decoders, what we do is basically a batch update to the existing data. So again, just a summary. And so what are the future things we're thinking about? One thing to point out is, as I think it's pretty clear from this conference, more channels, more data is better, right? And so one of the things that we found in B3 is that we can get really precise finger individuation and wrist control. And so what which is harder in B1 with 128, we could do it with a lot more data. This convergence confusion matrix is only a couple of days of data. And so we are definitely thinking about more complex hand control and not requiring a mode switch and so forth. So just to give you a sense of potential future directions. And really excited about future upgrades with channel density and so forth. So at this point, I wanted to switch to these other topics, all geared towards the idea of more robust control. And so one that we find kind of is a bit surprising, but has really helped with control. And it's this idea of slow fluctuations in internal states, right? And this is, again, related to complex robotic control in the environment. And as you'd imagine, the control can take 20 seconds, sometimes a minute. So these are long periods of time. And so, and what do I mean by internal state, first of all, right? External state is what we can observe, right? So, and you imagine a task, which we do, where the participants are waiting, and then we say, go, do the task, right? And so the green period is some long period of time when they're engaged. And the question is, well, what's the internal state, meaning how is the participant's brain viewing this? And one possibility, right, which is our assumption, is that engagement is just constant throughout, right? Even though it's 60 minutes, because a robot's moving, it seems to be doing well. Is it really that? Or to just foreshadow, it's really this very rhythmic fluctuations that's predictable that we can then leverage, and I'll show you that. And this is the work of Yasmin Graham and Hong Yeon, both bioengineering graduate students. Yasmin will be Dr. Graham in a week. She's going to graduate. And so one of the things that we're curious about is, even in periods of long transports, the velocity would fluctuate in a very rhythmic way as the inputs, because this is clearly related to some high gamma input to the system based on the decoder. So our hypothesis was that maybe there, particularly in the rise phase, there's a strong engagement, and there may be periods of disengagement that be underlying this. And importantly for us, why is this important? Because these low engagement periods are where the errors creep in, right? So the robot's moving, and these yellow periods, sometimes they'll drift off or do things that the participant didn't want, right? And so then they would spend time trying to correct those. So for robotic control, precision matters. So we really wanted to make sure we're only decoding when we think the participant really is engaged in that task, right? And so what we found was this interesting fluctuation of global high gamma and global beta, and I'll walk you through that. And as just more of a science experiment to explain the data, we had both participants do a really simple task where the robot endpoint's clamped. So all it can do is move to the right. It can't move off axis. It's clamped. And so they have to engage the task by imagining the right hand, the thumb once. And from then on, there's no engagement, right? So the thing is moving, no matter, even if they just stop thinking about it, it'll still move, right? There's no external feedback. And so despite that lack of feedback, we see these fluctuations, right? So this suggests these may be internal states that are fluctuating in a way, and I'll show you more about this. And it's super robust, right? So this is just the cross correlation between beta and high gamma, and I'll tell you more about what we mean by this. So the high gamma is simple, which is just the global average across the entire grid. So it's this global state of turning on, turning off. The beta is a little more complex and interesting. I'll talk about that next. And the red line is just a random shuffle. So it really just is really a true relationship. And so with beta, for those who don't know, beta's, it's been studied a lot. It's involved in event-related desynchronization, resynchronization. Has a strong signal in Parkinson's, in the adaptive DBS. I believe it's one of the control parameters. And so what we found was that beta is very variable. It depends on the area we're recording from, because we can take this brain and parcel it into premotor, motor, and so forth. So we found that we have to create the global state of beta by doing something called condition invariant beta, which is using demixed PCA, which essentially takes all the variants and you can marginalize into conditions or the whole full model. So condition independent collapses all action states into that. And when you do that, we find that the subspace spatially are this interesting area in premotor area. That's where the condition invariant beta is. And it maps onto things people have seen in EEG forever. It leads to ERD, ERS. But during active states, so this is now when the participant's controlling it, it's what's fluctuating. And so when we now look at this, and I should mention, the blue line is the raw data from our BlackRock system that we average across the grid. And then the red line is what we now will call slow beta, which is the 0.1 hertz. So we find if we do a power spectrum of this over long periods, it has a peak between 0.1 to 0.3 roughly. It's not a perfect oscillator, but 0.1 is a good fit for that for single trials. And so that's what this is, just a slow beta fluctuation. And so this is just a single trial example where during control, during transport, you see this interesting co-fluctuation. And if we look at a phase analysis, the phase of high gamma is roughly pi, which means it's anti-correlated. And we can look at a phase locking value, which is really looking at how well is the peak of one of the high gamma relate to beta, and it's quite robust. And if we do a random shuffle, it completely collapses it. So this suggests that these fluctuations are, and this I should mention is in the real world task, right? And or not real world, at least moving the robot and doing complex things, not the clamp task. And then what I showed you before was those fluctuations in velocity. And in fact, these fluctuations in global state really lock onto this global velocity peak. This is the speed, I should mention. This is mislabeled. And so again, the speed, when the robot's like really cruising, is at a low beta state, this condition invariant. And then as the beta starts to go up, the velocity drops. And this, again, has a strong phase locking value. And you start to really believe something when multiple features show the same thing. We see this with the velocity, with slow beta, high gamma, or the global high gamma. And then, interestingly, also with decoding errors, right? So this was really motivated. We want to make the robot sensitive and just ignore erroneous decodes, right? And so this, we give a very principled way to do that, where, so this would be at the start of a trial, you get this high global, well, this is not the global high gamma, but it's the decoding accuracy. So you get really strong accuracy, then it drops. So you can see this very fluctuating, accurate decodes. And if we were to actually ignore these peach or pink, whatever you call it, you actually get very high decoding, right? So it's very much better to decode the correct ones and ignore the ones that are not doing so as well. Because, as I'll show you, maybe fluctuations in engagement that are natural processes. And again, these are, if we randomly shuffle it, it kind of goes away. And so a natural question is like, well, what's going on with this, right? And so one of the things we did do was look at pupil diameter. And pupil diameter, as most of you know, is linked to sympathetic function. But during task itself, it's been studied a little bit. There's a paper from Bayer and Hughes group that shows in a BCI setup in a monkey, when the monkeys actually engage and disengage at fast timescales, there's actually changes in pupil diameter linked to sort of manifold engagement. So it's linked to sort of maybe accurate co-firing. And so we can see roughly anywhere from a 200 to 500 micron fluctuation in the pupil diameter. And we see a striking relationship where the pupil is actually constricting during this low beta state and will dilate, right? And so this, again, gives us a sense, we're not proposing to do eye tracking, but just gives us some confidence that these are really these microstates of engagement and then disengagement and so forth. And then when we look at this across time, we see very rhythmic fluctuations, again, with the periodicity of around 4 seconds, 8 seconds, right? So these are just inputs that are episodic that we think are changing the decoder output. Quick aside, this is not just about ECOG. So, with permission from Jose, I went back to look at the monkey data I collected, and we see very similar fluctuations, that even in a spike-based decoder that's continuous, the system is actually ebbing and flowing in a way that it doesn't look continuous. It's really this discontinuous input to the system. And so then the question is, well, how do we move forward, right? And this is still ongoing. And so, we can, the thing that worked reasonably well, we're working on others, a hidden Markov model, right? It's just a two-state system that is trying to label, so rather than saying it's all engaged, to go through and say it's really engaged, disengaged, and go back and forth, right? And so this is what we found is that through, and this is like a cool example where we see the high gamma go up, and you can see it decline, and then the beta, it's almost like homeostatic regulation between them. And so then we can label with this two-stage model disengage, and we're thinking about exactly how to do this, but for now we just do not allow inputs to the robot, and then it just sort of decays down and picks it up. And what it prevents is a lot of these erroneous type of decoding, which overall will improve performance, we think. So last thing we'll end on, a little bit more briefer, is again trying to think about how do we move all of this to more real-world tasks. And this is a topic called supervised autonomy with computer vision, and particularly focus on flexible object interactions, right? Not for like rigid autocomplete, trying to think about how in a complex environment can a BCI system interact with. And this raises, I got really read a lot about this literature for a while, and it's this idea of affordance, and this is from the psychology literature going back decades, even 50s. And what affordance is this idea that potential actions that an object offers to an individual is based on its physical properties and its most probable use, right? So our brains, and there's a lot of psychophysics on it, I look at my cup, and the brain assigns a probability I'm going to drink from it, I'm going to hold it from a lateral, right? And there's a lower probability of me picking it up from the top and so forth. And this has to do with pre-shaping and things that are almost subconscious, right? If you think about picking up a big glass, your hand will open up at a bigger aperture. If it's a smaller beaker, it'll be a smaller pen. And a lot of work, amazing work has shown, particularly the work of Leslie Ungerleider in monkeys, showed that some of these may be related to visual-based processing in these dorsal and ventral pathways, where the dorsal pathways are about what, parietal, like location, and the ventral pathways are about infratemporal object property semantics. And these are thought to be routed to, for example, PMV, the ventral premotor area, where there's a lot of known, where it's known to things like pre-shaping and grasp come about, right? So none of this is being incorporated into our system. And so this is something we've been thinking about, how to do this with computer vision. I should mention this is not new to us. Many others have thought about this. Anka Jurgan, Jen Collinger's group has published on this. And we've been thinking about how to implement this in our system and to do this in real-world tasks with flexibility. And you can think of this as a computer vision system that has a library of affordances, right? What's the probability of an object? What's the affordance, meaning, should there be a lateral grip or a top grip? If it's a bottle, maybe there needs to be an action to open it and so forth. What's the probable path? And then the question is how to integrate this with BCI control, the things that I've been talking about. And to do this, again, this is work of Sarah Seiko and Hong Yan. And so what we've been creating with feedback from our participants are real-world setups that are important for the participants. For example, Bravo 1, his number one thing is that sometimes he gets thirsty and he has to wait around for someone to come and ask him, am I thirsty? He'll get a water. It could be hours, right? So his number one thing is I want to be able to take my wheelchair, go to a fridge or a cabinet, get a cup, and get water for myself and drink it, right? So this is one of our focuses, and I'll just tell you that's something we've been working on. And of course, with a system that has full machine vision, computer vision, I mean, it could be trivial because it's knowing all the objects, right, and segmented. But what we really focused on is flexible actions, right? So our task would be get a cup from the right cabinet and get cold water, right, or opposite. So the way, our action is, our environments are predictable, but really the flexibility is in our sequence of actions, right? And that's what we wanted to implement. And to do this, I'll skip some details here, but we arrived at this, and Sarah Seiko, she's a roboticist, this system of supervised autonomy, and it's a subset of shared control, shared autonomy. And what it is essentially a handoff between BCI control and automated control. And it's all, the underlying process is a Bayesian inference detection. And it has an observation model, and this can be anything, right? It can be the joint angles of the robot, distance to objects, it can be neural features, and it's just trying to observe the intent of the user. But there's unassisted mode, which I've been showing you with the participants moving the robot. The system is trying to detect the most probable action, the most probable affordance, the most probable path. And why it's supervised autonomy is that the participant can always override it. It's not an autocomplete. And I'll show you some actions where the participant didn't like what it was doing, and it can set shift to flexibility. And so that's been the key that makes the participants really excited about this, is the flexibility and the ability to override. So it doesn't just, it's not like your Word document that formats in the way you don't want, right? They can override it and do a lot with it. And so towards this, as we built the system, it's worth thinking, where is the assist most needed, right? So if you look at all the data that I've shown you, the transport phase is quite easy for them. It's really, as you'd imagine, it's the object interactions, right? It's really hard to pick up an object, and especially when you don't have the visual feedback into our decoders. And so really the easiest thing to help with would be alignment, again, the grasp poses, and those are indeed what we implemented. And just give you a sense of, and so we've been working with our participants to figure out what objects do they care about. Things like tissues, things like cups, right? And these are extremely hard to do. It would take them 2 minutes to pick up a cup, right? And what's not clear shown here is like the trade-off. So there's a continuous trade-off between autonomy and the supervised autonomy. And I won't show you this, but the neural data we can actually decode when the participants are action observation mode or control mode. So we can start to use some of the neural properties into the Bayesian inference. And so then we've tried different control modes. And one thing that was interesting to us a little bit, you could imagine two types of supervised autonomy. One is a blended control, which is that the system says this is the optimal approach. And as the robot is being controlled, it just basically blends the optimal with the actual, so like an attractor. And we have used different blend modes like 0.9, which would snap to, or 0.1, and they really don't like that. They feel it's unintuitive, and it's almost like fighting something. And so the way, what they really liked is essentially a supervised autocomplete, which the system, when the system has enough confidence, it essentially takes over, but the participant can override it if and when needed. And it's interesting that they have learned to predict each other. So we can tell from the neural activity when the participant knows they need to subsume control back. And you don't see, it's really seamless. And so that's something we've been very excited about, incorporating the neural data into this autonomy as well. And this is an example of what override looks like, right? So maybe the system picked the wrong decoder, and then there's a switch that they can switch to and resume control. Whereas in the blended control, it's like really fighting a tractor, right? Then it's really overcoming it. It takes a lot longer. And then throughout this process, we asked the participants, is this under control? Were you able to correct errors? And using this feedback, it's clearly the supervised autonomy was the winner in the 3 systems we tested. So this is the last slide here. And so, as we move forward with this is the type of control that we are excited about, right? So this is, the machine learning is segmenting the environment, and we are, telling the participant to do something like get cup from the right cabinet and get hot water, right? And we can say, now open the drawer and do it. And one thing I should mention is that the camera is on the robot itself, so it can actually look into a drawer, right? Which is hard to do. And so you get to some interesting properties there. And this is something that, it's still work in progress, but it's, what's really improved is the grasp time, right? All of a sudden, that went from 60 seconds to relatively fast, something that looks almost natural. And so this is something where, it's still early days, but we're hoping to really build this library of actions based on their participants' needs. And they're surprisingly similar, right? B1, B3 gave us similar needs. And so it could become something generalizable with some customization. So just to summarize, what we find is that there's an unstable manifold, particularly in the early phases of learning. Accounting for this leads to long-term control, at least for 30 days. There's this interesting slow drift that we need to understand more after about 30 to 40 days, but a brief calibration will improve. And I think one thing to point out I didn't talk about is that drift will be more for really complicated tasks, like picking up a small object requires extreme precision versus a cup. And so that's something we need to think more about, like the stability really does scale or is really contingent on the precision required. And that's something we're working on. And then the supervised autonomy with vision seems to really help with the real world settings, but importantly remains flexible and enables autonomy still. So last but not least, just wanted to thank the Eddie, Adeline, and then folks in lab, Niklesh, Sarah, Yasmin, Hongi, and 2 other grad students. And then of course funders, the NIH, and then the Wild Neuro Hub. Thank you. Yeah.
Thanks. Thanks for the wonderful talk, Karunesh. Are there questions?
Hello, thank you for the great talk. You showed in a couple different papers that there was really no plasticity in the open loop neural representations, but that it all happens in the closed loop. Do you have any idea why they don't get better at imagery over time after having this BCI experience?
It's a great question, and I can go back to this. Thanks for bringing that up. Yeah, no, it's a interesting question, and just to kind of put what John Collinger was talking about is that if you look at this open loop spread, it's really constant. It doesn't change, and our hypothesis is it's a contextual. So these open loop, we don't have the screen on. It's a very straight. And so this may be a contextual that in the right context, I'm doing BCI control that can separate them. Is our best guess, because we just don't see that. So it may be that plasticity. I think is strongly contextual, and there's actually a really nice paper recently showing that in rodents that motor cortex doesn't change that much with learning. It's when animals are state 1 versus state 2, then the neural dynamics change really dramatically, and that there's coordination between premotor, and so I think that's our suspicion. These are contextually different, which is a good thing that if they're in different contexts, they can potentially come up with new control strategies. And it's interesting thing that also related to this is that the variance doesn't change. So the way, and we've worked really hard with them how to imagine, based on a lot of the work from Shinoy Lab and others, that what's your best control? Like it's curling your thumb in. So we really work in this, so they're imagining the same actions. Despite that, and I don't want to use the word sloppy. I think it's a feature, not a bug. That and then part of what we learned from the animal work we also do is that a big part of motor cortex and premotor is to shape variability. So a learned action is the same action with low variability, reliable feed-forward dynamics, and it may be tapping into that. Yeah, if that makes sense. Yeah.
Great talk, Greg. In the last section, when you're talking about the shared autonomy and the patient's overriding the current action, do they maintain all 7 degrees of freedom of control? And if so, if it's like in the middle of a grasping action, does the patient have to like undo the grasp specifically, or can it just move the endpoint backwards or something?
No, that's a great point. And I think partly what we have found is that they developed such a good internal model that they abort it well before the grasp. It's just in transport. And so this is what, this co-learning is not just about like decoders, but like the internal model. So they're really good. And it's almost, I guess if you'd watched this thing for a while, you'd know. So it's almost fixing the AI parameters are important because they know when does control come in, but also when it comes back. And So we can see like so we can see ramp up of firing rates before takeoff. So then it's just, there's no slowdown. So I think because they have a good model of that, then the trade-offs are seamless, and even the erroneous things it doesn't seem to bother them too much. They really like that, yeah. And then things that I was discussing with someone, there are really nice error signals in ECOG2, so the ways to do this even maybe prior to even conscious awareness, right? So that's something we're really thinking more about, how to incorporate into the Bayesian framework neural, not just behavioral, yeah. Yeah, thanks for yeah, Nick was a key part of the Bravo trial, right? With lots of hours of, yeah.
Hey, great talk. Thank you so much for this great work. I have like one clarification question and one comment. So the clarification question is that in ECOG signal, you are like seeing like something which in the initial phase is not very stable, the dynamics, right? Do you think that has anything to do with what kind of neural signal is being used?
It's a good question. I mean, I guess would we see this in spikes or just in different bands of ECOG?
I mean in ECOG, because I assume this data is from ECOG, right?
Yeah. So I mean, so we optimize so each channel gives you a raw trace, right? And we do subbands, and we found through some optimization that delta is really important, beta is important, and high gamma is 70 to 150. And what we do is, it's actually, we find that high gamma is super narrow band, and this Chang lab has also published on this, and then we just average all of those. And so probably beta is not that variable, it's much more of a global signal. So really, this is mostly in high gamma. And the real decoding is from high gamma and delta. We haven't looked too much on delta, frankly. This manifold drift is from high gamma. So it's possible delta is more stable. Yeah.
So like when you're using the signals here, so I'm assuming you're using only the sort of amplitude or analytic amplitude or something like that for high gamma signal or gamma signal?
Yeah.
So have you like looked in also using sort of phase or the instantaneous phase to sort of bring that into the decoder and try to make that work?
No, great question. So when we first started, we actually did these experiments where we took all features in a subband, including phase, and during closed loop adaptation, this is not nature biotechnology, and said with the loop, closed loop, if you put all features in there, which ones drop out of the network, phase gets completely shut out of it. So it's super noisy. It's really hard to estimate the phase in real time. So it's just too noisy, we find. So maybe there's other ways to do it, at least in our hands. Like the phase of beta is not that robust. The phase of delta is not robust. So So we picked high gamma, at least in our hands, high gamma, beta, and delta, because the weights of those grew over time and the others actually shrank. Because this closed loop decoding is basically it's weighting things that are predictable and unweighting things that are not predicting the kinematics, right, or the output.
Okay, that's a very interesting one. Sorry, I have one follow-up comment on that. So also like you observed some beta and gamma sort of anti-correlation is going on, right?
Yeah.
So I just have a comment that this kind of pattern is very much prominent even in the prefrontal cortex.
Yeah.
And we see that like very prominently, and it seems like that's a sort of some fundamental thing that how the sort of neural signals are at play, and that can be used in the decoder like this.
Agreed. No, agreed. I know of the Earl Miller work, beautiful work, and so I think the one thing that we found a little surprising is the time scale, right? So the Earl Miller thing is at like 3 hertz, super fast, right? And so it's like 0.1. So the time that's all. We academics, we say what's different, and so I would say I think that was a little surprising to us, this time scale of this interaction, 3 seconds, 3 to 5 seconds. And so the Earl Miller whole idea is about predictive processing. I don't think it's too far from what in beta may be either a pause or integration. We've asked our participants, are you aware of this? They don't, it's subconscious as far as we can tell. So I don't know how to map it onto that Earl Miller prefrontal. And they talk about LFP multi-layer, right? Feedback between layer 3. So I think it's just a fun, it's superficially same, but I've thought about it. It's not clear to me how to link it to that 2 area, right? Like layer 3, layer 5 output. I don't think it's that, because this is a global pattern, right? The entire grid is shutting up and down.
Yeah, like we can
Yeah.
talk about this like
Yeah,
outside,
yeah,
but
yeah.
thank you.
Yeah.
One last question, real quick.
Yeah.
Yeah, it's kind of a great talk, by the
Oh,
way,
thanks.
and following up on the kind of beta gamma oscillation, a two-parter. So one, is there any evidence that this is sort of like a local plasticity variation? So like, especially with the noradrenergic input, so if you like were to adjust the calibration on these local timescales, like would you see drift in the weights? Like, is the brain sort of exploring like, well, what if I try this? And then it, sort of trying to restabilize so you get the down state. And then relatedly, on the supervised autonomy, do you see changes in the beta gamma oscillation?
Yeah, no, great question. It's like we think a lot about beta, and I will say, like, this is not a plug for us, but just to say that towards your question, so this paper just came out this year. It turns out beta is actually an anti-plasticity signal. So we had animals learning and thing, right, and so low beta states. And not shockingly, high ripple states. And so when the animals transition to a more predictable pattern, that's when beta came up. So I think the beta, at least we find, is very similar to the Earl Miller idea of it's when we think there's an internal model. And that's one view of that. The other, it may be this idea of null states. So where it's seen a lot in motor cortex is pre-movement, post-movements, but also if animals make really fast movements and pause, you see really, and so one possibility is that, and this is using the parlance of the Chenoy lab, like movement potent, movement null. The system, it almost looks bistable. We have a work we're under review right now that the transitions between high firing states and beta really may be a way to have an effector quickly move and stop. And beta is super present throughout the basal ganglia. And when these are human participants want to cancel an action, beta goes up. So I would say consistent with the Parkinson's literature, not the predictive. So beta does a lot of things, but I think there's a predictive part, but also a null state that will hold an effector. And maybe what we're tapping into is is this ability to set shift. But I'm not sure it's plasticity because we have some data published that beta is, and we can induce beta artificially to the extent that you can, and we don't see learning actually. So something about this fast rhythm is anti-correlated with learning. And the other question was, with beta. So no, it's kind of really cool when we have neural activity of action observation. So when the participant knows that it's under supervised autonomy, that we can see beta go up when they know that they need to take over. So it's silent, and so this beta could be a really nice signal for latching to know when to turn on control, but also brief, like bistability, control, don't control. So there's a lot to learn there. And I think the other thing, I just want to point out that beta is not as simple as I'm trying to make it because of this condition invariant thing, right? So you have to, that beta does many things, and what I'm talking about is this area, this premotor thing, and it's consistent in both areas. I'm going to speculate here. I don't know if people have read the SCAN network paper recently, that in human, there's this area in central sulcus that is thought to be a somatomotor integration. And so when we look at our MRI and use like FreeServer to figure out where it is, this may be a node that is really sensory motor integration. And there's other betas, for example, an M1, PMV, that are much more about things that you're talking about. So, yeah.
All right,
Yeah.
thanks so much, Karunesh.
Yeah.